Chapter 5 Two Colors, a Million Smells. Man is a visual animal and has a hard time imagining any other way of being. We shut our eyes and picture of things from recent or distant pasts fill our minds. We dream, and our dreams are suffused with images, unreal though they may be, yet images surely. Our very grasp of whos and wheres and whats, from the concrete to the most abstract, is a grasp of visual forms and relationships. A fragment of a melody or a whiff of a scent may evoke a memory, but these are not normally the stuff human memories are made of, nor the stuff of human understanding. Yet dogs, like many other animals, inhabit a perceptual world where things are as likely to be understood and remembered by their smell as by their sight, where mental maps are assembled from avenues and topographies of odor, where the unseen is alive and vibrant and the seen is grayer and starker. If we could see through a dog's eyes, we would be shocked and dismayed by what had happened to our most precious link with the world about us. Detail lost, blurs that no amount of staring and focusing can alter, a world of washed-out hues and odd shifts of color. A dog could be equally appalled if he smelled through our nose. But dogs are not merely Mr. Magoo with a big nose. To see through another's eyes or smell with another's nose, we would need not only a nose or eye transplant, we'd need a brain transplant as well. Anatomical studies of a dog's eyes and ears and nasal lining can of course tell us something about the raw data he receives from his surroundings. Even before nerve signals reach the brain from the sensory organs, a lot of analysis has already taken place. Complex nerve cells in the light-sensitive retina at the back of the eye combine the pulses from individual light receptor cells to turn pixels into lines and shapes in motion. Nerves that line the membranes of nose perform an exquisitely sensitive sorting of odor molecules by chemical shape. But the real data crunching that yields a final, fully formed perception takes place in the brain itself, and the vast differences between the amount of hardware devoted to different senses in different mammals make it clear that there's more to smelling than a nose. A big nose doesn't just mean you smell a lot, it means you have a very different mind, too, one that inhabits a perceptual sphere that humans can only groupingly describe in words, never live and feel. Dogs and people don't see eye to eye, hear ear to ear, or smell nose to nose, all factors that explain at least in part why they don't always think mind to mind either. A dog's eye view. Like tourists who assume everyone speaks English, or should, it is second nature to us to think that the world looks pretty much the same to all creatures, great and small. We rarely give much thought to the optical process that turns light into vision. We assume that our visual version of reality is reality. Even those of us who wear glasses fall into this way of thinking. Glasses bring things back into focus so they once again look like they are. If those people who run around staging role-playing seminars on multiculturalism for business executives were to do the same for multispeciesism, I would suggest that the first group exercise they get everyone down on the floor with their eyeballs about 6 inches off the ground. Simply by virtue of visual perspective, the world looks very different to a Chihuahua. Dogs also differ from humans in their ability to focus on near objects, to perceive and distinguish detail, and to see contrast between light and dark. Some of these differences are relatively minor, but some must result in a highly altered version of reality. The most remarkable feature of the human eye is its extraordinary power of accommodation. The lens in a normal eye, when relaxed, is of just the right thickness and curvature to bend incoming light rays from a far distance, equivalent to the setting of infinity on a camera lens, so that they converge into sharp focus upon the retina at the back of the eye. If the lens were incapable of adjustment, the light rays from close objects would end up converging at an imaginary point well behind the retina. The result would be a grossly blurred image striking the light-sensitive cells of the retina. But by squeezing the lens with muscles that are under an unconscious control, we can make the lens thicker and alter its curvature, bringing close objects into proper focus. The greater the squeeze, the closer to our face is the focus. In young children, the eye's lens is capable of adjusting by as much as 14 diopters, an optical unit used in describing the power of lenses and in prescribing eyeglasses. That degree of accommodation corresponds to being able to focus on everything from infinity to an object less than 3 inches away. By way of comparison, eyeglasses with a power of 14 diopters would look like the Proverbial Coke bottle bottoms. Most glasses for corrective nearsightedness in humans run about 1 to 5 diopters. Dogs have a much more limited power of accommodation, generally no more than 2 or 3 diopters, which means they can focus on close objects only if they are no nearer than a foot or two. Anything closer than that will unavoidably be a blur. That may well explain why dogs generally try to sniff or touch objects at close range. They simply cannot see them very well. If the relaxed lens normally brings a distant object's image into focus behind the retina, the result is hyperopia or farsightedness. Accommodation can generally make up for the misfocus, but that means a corresponding loss of close vision. If the relaxed lens brings a distant object into focus in front of the retina, the result is myopia or nearsightedness. And in that case, distant objects will always be blurry without the use of a corrective lens, as accommodation can only move an image forward, not back, relative to the retina. Optical measurements of dogs' eyes have found a surprising incidence of myopia in some breeds. A study of about 200 dogs by veterinarian Christopher J. Murphy and his colleagues found the average canine refractive error to be pretty close to normal, within a quarter of diopter of a perfect, an amount that would not provoke any person to get glasses. Several breeds of sporting dogs such as Chesapeake Bay Retrievers, Golden Retrievers, Labrador Retrievers, Cocker Spaniels, and Springer Spaniels were on average a bit farsighted. But 2/3 of Rottweilers and half of German Shepherds and miniature Schnauzers in this study were significantly myopic by more than 1.5 diopters. The myopic Rottweilers were close to 3 diopters nearsighted on average. Generally, people who have more than about 0.75 diopters of nearsightedness will complain of noticeable impairment and find they need to wear glasses or contact lenses to function in everyday life. The animals in this study population were all pets. Interestingly, when Murphy and his co-workers looked at a second population of German Shepherds, animals kenneled at a guide dog for the blind in San Rafael, California, they found that the guide dogs had average normal vision, with fewer than thirds showing even as much as 0.5 diopters of nearsightedness. The guide dog program did not specifically test dogs' visions in selecting animals, but they did flunk out any dog that failed to perform well in training, which suggests that myopia results in a real impairment in getting the job done. The average farsightedness of sporting dog breeds suggests that there has likewise been selection at work in these breeds, that good distance vision has a demonstrable effect on making a good working dog. The researchers noticed a tendency for a severe nearsightedness to run in families, which suggests a strongly inherited component. In breeds that are not expected to perform anything more demanding than lying on the carpet, walking on a leash, and finding their supper bowl, there has no doubt been little selection for good vision, which has allowed myopia to sneak into the gene pool. There are distinct breed differences in peripheral vision and overall field of view as well. Human eye looks straight ahead, giving us about a 180-degree field of view, but with a lot of overlap between left and right eyes. Animals can see in true 3D vision only when they use both eyes together, and the overlap in human visual field thus maximizes the region in which we can perceive depth by using this binocular vision. The eyes of a dog are turned a bit to the side, which allows them to see a bit to the rear with a wider overall field of view, though at the cost of a smaller region of binocular vision. Dogs have a wider field of view than humans do, but the range of overlap of the two eyes is similar, limiting three-dimensional vision to a narrow sector. Shorter-faced breeds have a more sideways-looking eyes, long-nosed breeds more forward-looking eyes, but in long-nosed dogs, the nose literally gets in the way, which cuts down the total field of view of each eye, as well as it overlaps with the other eye. Long-nosed dogs especially have a problem when they try to look with both eyes as anything below the horizontal plane of vision. Dogs generally do just fine with their more limited 3D vision. The times they really need to perceive depth accurately are when they are looking straight ahead at a single item of interest, rabbit, frisbee, and so on, and that is well within the binocular field of view of almost all dogs. Studies on puppies have also shown that even with one eye covered, they can pick up depth cues that allow them to make pretty accurate judgments about how far away an object is. Shadows, relative motion, relative size, and other features of objects all convey depth information that the dog brain can process and draw deductions from, with or without binocular vision. The ability to pick out detail at a distance depends in part on the ability to focus sharply, but It also depends on how fine-grained the light sensors in the retina are. In general, the more densely light-sensitive cells are packed into the retina, the greater the ability to resolve small details. It also matters how these cells are wired up. Dogs have excellent night vision, but one way they achieve this is by ganging multiple light receptor cells together and coupling them to a single ganglion, the nerve cell that transmits the resulting signal to the brain. The result is exactly analogous to what happens in high-speed camera film. Each grain in the film is more sensitive to light, but each grain also covers a larger area, resulting in a coarser-grained image. In primates, one light receptor cell plugs into one ganglion. In carnivores, such as dogs, it is typical to find 4 light receptors converging on each ganglion. Overall, the density of ganglion cells in dogs is considerably lower than in humans, and the total number of nerve fibers running from the eye to the brain is about 170,000 in dogs versus 1.2 million in people. The highest density of ganglion cells is found along a horizontal streak that runs across the center of the retina. This is where an image from the distant level horizon would fall when the eyes are looking straight ahead, precisely the situation in which picking out fine details is most needed. This visual streak is much less pronounced in many breeds of dogs compared to wolves, however. The visual streak in wolves has about 14,000 ganglion cells per square millimeter. Some dogs equal that, but others have fewer than half as many. There are several ways to measure the acuity of an animal's vision. One method is to train the animal by presenting it with 2 cards, one containing alternating black and white stripes and the other a solid neutral gray, and rewarding it for picking the striped card. Once the animal does this reliably, it is then given a series of choices in which the stripes on the black and white card get narrower and narrower each time. When the stripes reach the point that they blur together in the eye into an undifferentiated gray, the animal's chance of picking the right answer falls to 50-50. A similar experiment has been performed by monitoring the animal's brainwaves, as it is shown increasing narrow stripe patterns. When the blurring together point is reached, the brainwaves show a characteristic change. Normal humans can pick out a stripe that fills as little as 1/60th of a degree of arc in their visual field. Dogs cannot see anything narrower than about a 16th of a degree of arc, which corresponds to 20/75 vision. What a normal human can see at 75 feet, a dog would need to be 20 feet away to see. The sharp-eyed blue jay, by comparison, has about 60-20 vision. It can see at 60 feet what a normal-sighted human needs to be 20 feet away to see. Dogs have made other trade-offs between sharp vision and night vision. Like many other primarily nocturnal animals, dogs possess an unusual layer of cells at the back of the retina known as the tapetum lucidum. These cells act like a literal mirror, reflecting incoming light back out through the retinal cells, giving them, in effect, another shot at detecting each incoming photon of light. But this process inevitably means further blurring, at least in dim light. The tapetum is also why the eyes of the dogs, cats, horses, deer, and many other animals glow so brightly yellow when caught in a flashlight or headlight beam. The red eye in humans, that is the bane of flash photographers, results from the less intense reflection of light of the blood vessels at the rear of the retina. There are some interesting hints that the tapetum may not be a completely passive mirror, and that its particular chemical composition may cause the color of incoming light to shift slightly as well. Blue light is absorbed by riboflavin molecules in the tapetum and then re-emitted at a color closer to the center of the spectrum. These re-emitted colors coincide with the wavelengths at which the eye's black and white receptor cells, known as rods, are most sensitive. Thus, the blue-black of the dark evening sky may well appear much brighter to a dog than to us. This would exaggerate the contrast between dark sky and dark objects on the ground. Only a few studies have been made of how the dog's higher-level visual processing machinery puts together data from the retina to form a perception of shapes and motion. Studies in which dogs are trained to discriminate between shapes have found that they can rapidly learn to tell horizontal lines from vertical lines, but have a harder time with more complex geometries, such as upright and inverted triangles. Dogs are acutely sensitive to motion. However, in one test, a stationary object that dogs had to be within a quarter of a mile of to notice could be spotted in motion a half a mile away. Canine color blindness. Early studies of color vision concluded that, with the exception of primates, most mammals, the dog included, could not perceive color at all. This is still an apparently widespread belief among dog owners. However, careful studies by Jay Neitz of Wisconsin Medical College and his colleagues have established definitively that dogs do possess color vision, albeit of a limited kind, similar to a form of color blindness in humans. Humans are able to distinguish the full spectrum of colors in the rainbow because they have 3 different sets of color receptors in the retina, each with a peak sensitivity to a different wavelength of light. These 3 wavelengths correspond roughly to yellow, green, and blue. When light strikes the retina, the brain, in effect, compares the relative strengths of the signal coming from each of these 3 types of receptor cells, known as cones, to obtain a precise fix on its wavelength. Light that stimulates the blue and green cones equally, for instance, is perceived as blue-green. The combined response of the yellow and green cones sorts out whether a color is red, orange, yellow, or green. Red light, for example, would stimulate the relatively nearby yellow cone much more than the green cone, farther down the color spectrum. Dogs, and in fact most non-primate mammals, have only 2 kinds of cones. In the dog, these cones have their peak response at wavelengths that correspond to the colors of yellowish green and violet. That means that while the dog can see both red, orange, yellow, green colors and blue-violet colors, and can tell that any of these colors are colors as opposed to white or gray, they cannot tell the colors apart within these 2 broad groupings. They cannot tell red from yellow or green. They cannot tell violet from blue. In other words, in contrast to the approximately 100 distinct hues that humans can discriminate in laboratory tests, dogs can distinguish but 2. Intermediate colors in the dog spectrum, those in a narrow band in the vicinity of bluish-green, would not look like any colors at all to the dog, but would rather be indistinguishable from white or various shades of gray, since these medium colors would stimulate both types of cones equally, just as does white light. This narrow, colorless band in the middle of the spectrum is known as the neutral point, and it occurs only in animals that have only 2 kinds of cones. Neitz was able to demonstrate color vision in dogs by training 2 greyhounds and a poodle to nose display panels on which colors were projected. The dogs were given a choice of 3 panels and were rewarded for nosing the one that looked different from the other 2. It was a straightforward, if time-consuming task, to show that when all 3 panels were illuminated with white light, the dog could usually tell when a specific colored light was added to one panel but not the other 2. But when the colored light reached the vicinity of bluish-green, the dog's ability to detect its presence plummeted quickly to the level of chance, and when 2 colored lights equally spaced on either side of the neutral point were blended together, the dogs also failed to distinguish that mix from white light. A practical consequence of Neat's findings is that many of the things we make for dogs are the wrong color. A bright red-orange dog toy stands out dramatically against the lawn to us, but to a dog its color is not readily distinguishable from the green of the grass at all. A violet object would probably be a much better choice when the background is green. Trying to teach a dog to pick out different colored objects is probably going to be a losing proposition when the colors are, say, red and yellow or orange and green. Likewise, it is probably impossible to train guide dogs to tell apart the red, yellow, and green colors of a stoplight on the basis of color alone. Dogs, like most mammals, probably lack full color vision as a result of evolutionary pressure that go back tens of millions of years. When mammals first appear at the end of the dinosaur age, Neitz points out the only niches available were nocturnal ones, so the first mammals had a need, above all, for night vision. The cones that provide color vision are not, however, very responsive to dim light. The much more sensitive rod cells in the retina, by contrast, cannot discriminate colors at all. So the trade-offs in the early mammals favored giving up some cone cells for more rod cells. The 3-color vision in primates was apparently only reinvented much more recently as an adaptation to their special place in nature. Wolves, being essentially nocturnal, still gained more from having a preponderance of rod cells to boost night vision than they lost from having a lack of full-color vision. They also appear to be able to distinguish shades of gray that are imperceptibly different to the human eye. The two-color visions of wolves and dogs provide some insurance against being too easily fooled by camouflage, which would be the fate of an animal that could see only in black and white. Black and white camouflage has not only matched the overall brightness of the background, not color at all, but beyond that, it is safe to say wolves lack full color vision for a simple biological reason. They simply didn't need to get the job of being a wolf done. Sharp ears. Differences in hearing ability also strongly reflect adaptive need. The legendary ability of the dog to hear high pitches has been verified by laboratory tests. Dogs can hear sounds up to about 65,000 hertz or cycles per second. A healthy teenage human being, or to be more precise, one who has not already blasted his ears or out listening to a Walkman at full volume, can hear sounds up to a maximum of about 20,000 hertz. To produce the top note a person can hear would require adding about 28 extra keys at the right-hand end of the standard piano keyboard, or about 2 1/3 octaves to get to the top dog note. Would take 48 keys or 4 full octaves. Dogs do not themselves produce any sounds this high, but small rodents do, and thus their hearing range may reflect a predatory adaptation to finding high-pitched things to eat. Dogs, like most predators, also possess a fairly precise ability to tell the direction a sound is coming from. Humans can tell which of two possible sources a sound is coming from when they are separated by a little as 1 degree of arc. Horses, cows, and goats have trouble figuring this out even when the sound sources are separated by as much as 20 or 80 degrees. Dogs do well down to about 8 degrees of separation, on a pair with cats, ferrets, opossums, sea lions, and monkeys. Sound localization is accomplished by a combination of acoustical physics and brain computation. In general, the brain calculates the answers by comparing the relative loudness of the sounds reaching the left and right ears, or by measuring the time delay between the arrival of the very start of a sound at each ear. A big head helps, because it maximizes the distance between the ears, thus increasing the difference in arrival time and volume between left and right. But a lot clearly depends on the brain circuitry. At some very large animals, such as the horse, are very bad at sound localization, while relatively smaller-headed animals, notably man, are quite good at it. Dogs obviously came equipped with more advanced circuitry needed to do this well. Careful measurements have shown that the sound localization ability of dogs correspond to an ability to distinguish arrival times in the left and right ears as little as 55 microseconds. Dogs and humans possess another sort of sound localizer that relies completely on brain wiring. One difficult problem in sound localization is what to do about echoes. A sound coming from the right might bounce off a tree or wall to the listener's left, creating a reflected sound wave that arrives at the ears from the opposite direction from the waves that arrive directly from the source. Human infants up to about age 4 months are confused by such echoes and are unable to tell where a sound is coming from when it is accompanied by reflected sounds. But older infants have no trouble turning towards the actual source of the sound, even when there are reflective waves coming from a confusing direction. Studies have shown that the brain circuits actually shut out the delayed signal of the echo. The brain, in effect, can recognize that this second signal, which arrives a millisecond or more later, is an echo, and actively suppress it from notice. Studies in dogs have shown that this ability to filter out reflected sounds appears during the course of development in puppies, too. The calculus of olfaction. Like the eye, the nose is both sensor and computer. The eye's retina is actually an extension of the brain. In computer terms, it is a network's PC, not just a dumb terminal. The primary nerve cell of the retina that respond to the individual's pixels of incoming lights are wired to a series of more complex nerve cells, all still within the retina, that begin to do the job of making sense of more complex geometric patterns like lines and shapes. The nose is a networked PC too. A large bundle of nerve cells known as the olfactory bulb resets directly above the mucous lining of the nose. Its full-time job is processing the smell signals relayed by the primary receptor cells, whose long, thin filaments extended directly into the mucous lining of the nose. The olfactory bulb in the dog is dramatically larger than it is in the man. The canine nose also has something like 20 times as many primary receptor cells as the human nose. How all of this works to detect odors is one of the great scientific wonders of the world. Studies in a number of species have found that different regions of the mucous lining within the nose have different chemical properties, more readily absorbing chemicals of one particular molecular shape or another, or preferentially absorbing in one region chemicals that are more water soluble and in other chemicals that are more fat soluble. The ability of the nose to make precise chemical distinctions is truly extraordinary. Some pairs of chemicals that exist in nature are identical in every way. They're made up of exactly the same elements joined together in exactly the same three-dimensional sequence, except that one is the three-dimensional mirror image of the other. Yet such stereoisomers frequently have a dramatically different odor, indicating that the nose can sort them out by their complex shape alone. The molecule carvone, for example, has the odor of caraway. In one of its stereoisomers, the odor of oil of spearmint in its mirror image form. Measurements of the acuity of the dog's nose suggest that the dog is many times more sensitive than man to the presence of minute quantities of odor molecules wafting in the air, but the data are all over the map. This is probably in part because the threshold for detecting different chemicals no doubt varies dramatically according to the particular chemical involved. Some comparative studies have found that dogs can detect certain organic chemicals at concentrations 100 times less than people are able to. For other compounds, the dog's edge may be a factor of a million or more. In police and security work, dogs can detect the odor from natural gas leaks, concealed narcotics, explosives, and currencies, all at levels well below the threshold at which humans are aware of the odor. In controlled studies, dogs could detect human scent on a glass slide that had been lightly fingerprinted and then left outdoors for as much as 2 weeks, or indoors for as much as a month. They could pick which of 6 identical steel tubes had been held in the hands of a person for no more than 5 seconds. They could distinguish between t-shirts worn by 2 identical twins who ate different foods or by 2 non-identical twins who lived in exactly the same environment and ate exactly the same foods. More than such a remarkable sensitivity to trace odors, it is the ability to pick out particular odors of interest from a welter of competing smells and to match and distinguish them that is the dog's most impressive olfactory feat. This ability is surely a reflection of the dog's superior olfactory computing powers, for it requires not just smelling but analyzing. Dogs have no innate interest in the smell of people's narcotics or $100 bills, but if trained repeatedly to focus on certain categories of smells, they can perform mind-boggling feats of cross-matching. In the twins and t-shirt experiments, the dogs were first trained by presenting them for 15 seconds with a simple t-shirt doused with an artificial smell. They were then allowed to approach a small trough 10 feet away and fetch whichever of the 2 shirts placed there matched the sample. The experimenters found that the dogs did better when the 2 t-shirts were placed fairly close together in the trough, 1.5 feet from one another, which permitted the dogs to smell the 2 simultaneously. In the actual test, the dogs were given as a sample a shirt worn by one of the twins for one 24-hour period and had to pick from the trough the shirt worn by the same twin during another 24-hour period. With a lot of practice, dogs can perform the even more difficult task of matching the odor from one part of a person's body to another part, pants pockets to hands or crook of the elbow to hands. When presented a sample odor from a person and then given a choice of 6 different tubes, one of which had been rubbed on a different part of the body by the same person, trained dogs did significantly better than chance. but they made a lot of mistakes too. The average performance for the elbow and hand cross match was about 1 in 3 correct, versus 1 in 6 expected by chance. On the other hand, when the suspect was a person already known to the dog, the correct response rate shot up to 73%. Part of the difficulty dogs have with this task is that while person A's hands smell different from person B's hands, person A's elbow also smells different from person A's hand in some ways. At the same time, some odors are common to all humans. The dog is clearly capable of detecting and distinguishing all of these smells, and the challenge is for the dog to learn which comparisons are the ones its seemingly fickle human handlers are interested in. They are not necessarily the comparisons of innate biological interest to the dog. An extraordinary combination of sharpness of nose and olfactory computation is also involved in the amazing ability of dogs to find and follow tracks. A series of simple but brilliant experiments by Norwegian and Swedish scientists have provided some wonderful insights into how dogs perform this task, which probably seems the most incomprehensible to us of all their olfactory feats. The scientists were inspired in part by a 13th century Icelandic saga that tells of two Norwegians who, attempting to elude their Swedish captors, tie reindeer hooves backwards to the soles of their boots, intercepting their trails. The Swedes' dogs follow in the wrong direction, ending up at the empty hole in a pigsty where the prisoners had been held. The scientists first tried the same ruse, having a person walk either forward or backward across pavement or grass. 20 minutes later, a trained tracking dog and its handler, neither of whom knew which way the person had actually walked, were allowed to approach the midpoint of the track, and the dog was set to work. The dogs consistently were able to follow the trail in the actual direction the person walked, regardless of which way the toes and heels were pointing. So much for Icelandic sagas. The dogs' performances were videotaped, and the dogs were also equipped with wireless microphones taped to their noses to record their sniffing. When the scientists reviewed the data, it was clear that the dogs had almost no trouble making up their minds which way to go. In fact, they made virtually snap decisions. The dogs would sniff from 2 to at most 5 of the footprints for a total of 3 to 5 seconds, then strike out in the correct direction almost every time. Further tests confirmed that, as incredible as it seems, the dogs were apparently able to determine which of the footprints had been left the most recently by the strength of its odor. The odors the nose is capable of detecting are all by definition volatile. That is, they are the product of chemicals that evaporate into the air and thus can be inhaled into the nose. Thus, the older the track is, the more its volatile components have already evaporated, and the lower the concentration is that remains. The scientists found that the dogs could readily detect and follow the trail left by a bicycle, but were unable to determine its direction. But when they fastened leather strips around the rear tire of the bicycle, thus laying a trail more like footprints, discrete patches of scent at spaced intervals whenever the leather strips struck the ground, the dog once again were able to determine direction of travel. Again, that supported the notion that the dog is making a direct comparison of relative strength and thus relative age of the odor from 2 or more adjacent prints. The continuous track of the bicycle provided too smooth a gradient of odor strength to allow such comparison. Finally, the researchers tried smearing sausage on the leather strips, the clever idea being to leave a scent track that would be an odor version of the Norwegian prisoner's false trail. As the sausage wore off, it would produce an increasingly weak scent. Thus, the strongest smell would be at the start, the weakest at the finish, the opposite of what happens with a normal trail. Sure enough, the dogs were consistently fooled and tracked the sausage-modified bicycle the wrong way. Dogs generally find it impossible to determine the direction of a trail more than 3 hours old, even if they can still follow the trail. That suggests that the overall odor has faded to the point that the difference from one print to the next is just too small to discriminate. On a 30-minute-old trail left by a person walking 1 stride per second, the relative age of 2 adjacent footprints differs by 1 second out of 1,800 seconds. Assuming smell fades at the constant rate, that means the dog's nose must be capable of detecting a difference in odor strengths of at least a like ratio. 1 part in a couple of thousand. That boggles our minds, but by way of comparison, the human visual system is perfectly capable of similar feats of discrimination. If you hold 2 needles out at arm's length, one above the other, your eyes and brain cell can tell if one of the needles is a fraction of a millimeter closer to your eyes than the other. This is less than part in 1,000. It is all a matter of what the brain is wired to do.